Through our trials and working with growers, we know that controlling weeds is a real challenge. And a lot of that is to do with the germination pattern of those weeds and how that affects their control strategies. So we're here today, just south of Cambridge, at one of NIAB's trial sites, looking at some research they're doing into germination timing and the relative competitiveness of those weeds in a crop. I'm joined by John, who's going to tell us a little bit more about the trials.
This is a trial where we have a whole range of different weed species, and we're looking at what is the consequence of those species' germination timing on the potential seed return of individual plants in the population. One of the challenges of grassweed management is that different weed species have different germination timings. So the consequence when we come to apply a herbicide is that we have a whole range of individuals at different growth stages and sizes. So our herbicide control timing means that those different individuals are differentially controlled. The survivors have different levels of potential weed seed return. So as an example, I've just walked around the commercial crop here and found a wild oat plant, which is just a seedling that germinated just a few weeks ago perhaps, and then a much larger plant which probably germinated in the autumn with the crop. Now obviously one plant has a very high potential seed return, much higher than a spring emerging individual. A larger plant is much harder to control than a smaller plant. So if you wait until the spring, wait until every last possible plant has emerged, we may be able to control the whole, get herbicides on the whole population. But these much larger plants with a much higher potential seed return are not going to be as well controlled. If you apply your grassweed herbicide much earlier, you may well miss these small individuals, get better control of these large individuals. And this trial is just trying to quantify what is the consequence, what's the potential seed return of these much smaller, later germinating plants compared to these earlier germinated plants. And then obviously in a field trial we're able to manipulate and introduce a whole range of really contrasting species. These wild oats, winter and spring wild oats, one of the brome species which is increasingly problematic, but also things which are much more autumn dominated germination weeds like blackgrass and Italian ryegrass in one really quite neat small footprint.
So a better understanding of this relationship between germination timing and the size of weeds should help growers to adapt their control strategies by targeting their applications better and get better overall control. Is that part of what we're doing?
Yes, absolutely. I mean, some crops have got very limited options post-M, maybe only one opportunity, one application timing, and really understanding how these different species, how the individuals within those populations are contributing to seed return, driven by their germination timing, is how we're going to optimize that application timing for different weed species in those key crops. In our field trial, what we've actually done is germinated individual weeds in the glasshouse and transplanted them into the plots to give us that range of weed species and range of germination timings.
One of the things about germinating weeds in the field is it's notoriously variable in getting their germination. So to counter this for this trial, what we've done is we've germinated seeds in controlled conditions. We've planted seeds at various times throughout the year to get them all to the same stage when we're planting them out in the field. So what we've really aimed for is something around this type of size, where we're just into true leaf stage. We've planted these seedlings out at 5 stages throughout the season, 2 in the autumn, in October and November, and then 3 in the spring. So this represented some time in March, April, and then 1 more in May. Once these plants have grown, we will then begin to sample them. This sampling is to really capture the biomass of each individual plant to see how the effect of the crop throughout the season has had on its final size. There'll be 3 sampling timings in total. The first 2 will be tied into the key fungicide timings T1 and T2, which are often times where growers will be using herbicides at the same time. The third sample timing and the final sample timing will be to look at both biomass, but also the seed return for each of the 5 species that are used in this trial.
So John, it's the middle of April, we're out in the field. What can we actually see?
Well, we're standing in the winter wild oat bit of the plot, and we've got our 4 germination timings. We've got some plants that germinated in October when the crop was drilled, some in December, some in February, and then some in March. And you can see we've got these much larger individual plants for the earlier germinating plants. They're well tilled, we've got quite a lot of biomass. And then as we go across the plot, we've got some of these much smaller December germinating emerging plants. And then we go across again, we've got some from the February timing in here.
That's it, yeah.
And then of course the smallest plants of all are just a month or so old, much smaller.
Okay, so in theory, this could be an appropriate timing for applying a herbicide in this crop. And actually growers could be looking at targeting weeds which are like those first ones we looked at, which germinated with the crop, really well tilled, and also some very recent germinators. So in terms of rates of herbicide for control, you'd need to be adapting that strategy, wouldn't you?
Yeah, so you want to be making sure that you're picking the appropriate dose for the largest individual in the population that you're trying to control. I mean, obviously we have a whole range of weed species and these different germination timings in this trial. For the grower, it's really important that they understand the nature of the grassweed problem that they're trying to control. What is the species? What is the mix of individuals all germinate at different timings that they're trying to control and get the rates right? And then obviously the application timing is absolutely critical to optimise over time.
So in terms of what we'll do next, we will see the results of this work published later. And yeah, thanks for joining us.